Hello, my name is Rushikesh. I'm from Croda, and today we are going to speak on biological formulations. Nowadays, very much inquiries are being received by us. We are seeing that people are more and more working on biological formulations. These type of formulations are getting importance nowadays, and therefore we thought that we'll speak on biological formulations. We'll brief you what Croda is having in their basket, how Croda is approaching to these formulations, and how we can support to our customers for developing these kind of formulations. So next 20 minutes, I'll speak on how we can do this, what we have done, which products of Croda or from Croda basket can help you to develop these formulations. And then we can speak in future for developing these formulations or our support, Croda support for developing this formulation to our customers. Along with me, there are my colleagues Om Dande, Shirish Ingrade, Dhananjay Kumar, Rohit Pangetkar, and Vaibhav. These are sales colleagues which are faced to customers, and you can approach them for any support. We have our marketing colleague Rashna, who is helping us to recording this formulation so that we are reached to you. We have technical team under guidance of Dr. Rahul Hedav and other colleagues who are helping our customers to make the formulations, and definitely in this particular formulation will definitely help you to develop the formulation as per your requirement. We know that there is various formulations which is under biologicals, but we will be focusing more on microorganism based formulation. But there are like different extracts or essential oils or biochemical formulations are there, or based on those there are formulations. But today we thought we'll speak only on microorganism based biological formulations, but we have options for other types also. So if you have some other requirement on essential oils or extracts of plant extracts, etc., we are always there to support you. So how Croda will support you in making this, or why people are going towards this kind of formulations? This is sustainable formulations. That's what people feel. Not always that all natural formulations are sustainable or are safe, but still people feel that some of these are really sustainable and safe formulations. For organic agriculture market, people are going away or moving away from chemical pesticide based formulations, and therefore biological formulations are in demand. Registration of biological formulations or biological control agents based formulations is easy. In some of the countries, it's easy to register these kind of formulations, obviously versus chemical pesticide formulation or chemical based pesticide formulations. There are some advanced technologies are coming up, and therefore biological formulations are in demand, and therefore people are going more and more towards this kind of formulation. Some of the chemical pesticide manufacturers are also aiming for this niche market. And then rapid growth in seed treatment market. You know that Croda has got a company in Incotech which has got seed treatment. So if you need anything in seed treatment, we can help you out through Incotech. Now, how one can approach to biologicals? As I mentioned earlier, the conventional products, the chemical pesticide based formulations, definitely they are going to continue. At any case, biological formulations cannot replace conventional products. That is for sure. But conventional products and biological agents or biological based formulation, if they work together, that can be a part of integrated pest management program. And today, if you see that all are not convinced that biological control agents could be very good in future. But then still people are working, and not everybody, but some people are really working on this. The major challenge in biological formulations or biological control agents based formulations are cost, manufacturing, formulation stability, etc. Some of the major chemical pesticide companies, they are also looking for niche market by using biological formulations, or some startups are there who are gaining market access and hence more focused on biological formulations. Now, what Croda's activities are in this type of formulations? What we are doing, we're trying to understand the market, how it is moving, what are the gaps, what is the requirement of our customers. We're trying to understand that. We are collaborating with research institutes, experts in biological formulations to understand how we can work together with them. We are studying our portfolio. We have few products in our basket which are only certified products, and we are working on those products or studying those products with various microbes, trichoderma or pseudomonas, etc. We are seeing regulatory approach, what it is, how we can approach to regulatory with the data, what we have generated with the help of our customers. And then obviously we are expert in formulation development. So we are developing formulations of various types for biological formulation, especially microorganism based formulations. And then evaluation of application in seed, that's again part of Incotech strategy, which we are working on. So in short, working in collaboration with customers or partners for solutions to give a sustainable agriculture. So that's what we are focusing. But you must have noticed that I'm not saying anything on development of microbes. See, development of microbes is never Croda's focus because that is your area of interest or your area of expertise where we do not want to come. But once you develop some microbes, we will be there to support you to convert that microbes formulation of microbes in a stable formulation which will give stability for longer time, better efficacy, shelf life, etc. So we will be working with you on those microbes. We'll never develop microbes on our own. Now, as you know that Croda is a company who is focusing always on sustainability. So I'll just brief here what we are doing on sustainability or achieving those goals. We know we are having eco range products, 100% bio-based or renewable based products, where we are giving these products. For example, Twin series is again bio-based or renewable ethoxylated based product, bunch of products, and then it's always available for our customers. Biostimulant Plant Impact, it's another company of Croda. We have biostimulants in Plant Impact basket for especially for cocoa, and we have seen that it increases the yield of cocoa by 40% on an average basis. Seed treatment, Incotech developed coatings for seed that has got benefits of traditional seed coating, and at the same time it doesn't contain any microplastic, so that will help to save the microplastic or reduce the microplastic, and obviously it will help to marine life. Biological formulations, again working together in collaboration with our customers with microbial knowledge and our formulation expertise, and together we can work on these type of formulations and can deliver a solution to the market as per the market requirement. Now, what microbial facilities or expertise we have in our Croda laboratory across globe? We have some microbiological methods where we can generate high definition images, microscope images, or in vivo microbial testing. We can do seed treatment application. We can do what microbials or biological viability or recovery screening, etc., etc. We have this kind of microscopes across 4-5 laboratories globally, including what is at Thane. We can do physicochemical characterization, formulation behavior, or characterization of formulation under environment conditions. That capabilities we have in our basket. Few of you have visited our laboratory, so you know what facilities we have at Thane. Improved purity. Few of you know that Croda is well known for super refined products. Basically, these products are going for pharma sector, healthcare requirement, and these products help to improve the shelf life of active pharmaceutical ingredients. So we thought that we can have some high purity or improved grades of surfactants, which can help to reduce impurities in microbes and impact on viability would happen definitely. So those customers who would like to work with us where their microbes are not stable, shelf life is less, definitely we can work with them to give improved version of surfactant or purified version of surfactants, and then we can work together to find out how that is helping to impact to improve the shelf life of the microbes, or how it will impact the stability of the formulation or efficacy of the formulation. Now we have a global structure, we have qualified team, we have microbiology team, we have formulation laboratories across globe, and this in combination would work with customers, and we have some specialists in these areas we can give who can give solution to our customers and can develop a very good product in biological formulations. So I'll just go to formulation microorganisms. What we have done now, what are the challenges in these formulations or developments? So if you consider in the formulation, this includes a shelf life of the end formulation, handling and application of microbes, of enhancing bioactivity. So we have to ensure that active ingredient reaches to the target after spraying, protection against external factors, so maybe temperature conditions. Some cases microbes are sensitive to moisture, so those are external factors. We have to give protection against that. Maintaining good distribution of microorganisms. When you dilute the formulation for spraying, how you can distribute the microorganisms, that's important here. And of course, no or low chemical residue in the field. So these are the challenges when we do any formulations in this area. Now, development of these formulations for microorganisms involves several steps. That how is your active ingredient? What is the formulation selection? How will you select the formulation? How you do the dilution, or how you can do the application technology if it is seed, or how you control the target when you spray the formulation or diluted version of formulation, how it will go to the target. So these things would need to study while developing the formulation, would need to be considered while developing the formulation. So when you have any active with you, so you need to understand how this active ingredient is or how it behaves. Now, same species may have different stains which may show different performances. External stresses like temperature conditions or moisture can affect the performance of that strain. Depending on target, one strain may perform better, another may not. Morphological structure of microorganisms, which is extremely important, and then we'll see how is important in next slides. And then strain, we have some own peculiarities which we need to address while developing the formulation. Now, morphological structure of microorganisms can be extremely hydrophilic or hydrophobic, depending on structure, and then based on that you have to decide what formulation you have to develop, how we are going to dilute that. So these are to be considered. Some of the biocontrol agents like trichoderma or Viability type, these are grown and used in Conidia forms, and then you need to understand how one can develop a formulation. Then you need solid formulation, liquid formulation. If it is liquid, then oil-based or water-based, and then studying this further: stability, dilution, shelf life improvement or increase in shelf life, efficacy improvement. So all these things need to be considered or studied after development of the formulation. Now, selecting right formulation is important here, and when you select this right formulation, then you have to study thermodynamics or phase behavior, solution chemistry, surface chemistry, colloids, emulsion, suspension, etc. So again, based on what you are basically doing, it oil-based or solid-based. And if it is oil-based, then you can see here pictures. In one case, you can see a uniform distribution. In another case, distribution is not uniform in the formulation. So using ingredients to stabilize the formulation is important, and then we'll study further on physical chemistry of this formulation. WP type formulation, again, if you have a powder microbes, and then you can mix them with inerts, solid inerts, and then they can present benefits like simpler production process because it is WP. Spore germination would get reduced because of water-free atmosphere for storage. Since it is water-free, preservating agent need not to be added, and hence we get better stability or non-harmfulness to the microorganism. A storage sedimentation will not be there because that will happen in OD type of formulation. WP is low cost type of products, so that's the advantage. But again, limitation is dilute to difficult to mix in spray tank because we have WP and then adding in spray tank distribution is difficult. You need to use particular dispersing agent to uniform distribution of that. Dust formation would be there in WP formulation. So these things need to be considered while developing WP formulation of microorganisms. OD type of formulations, again, these are easy to apply comparatively. Since this is water free, no need of addition of preservative, reduced pore micro germination issue would be there. You can add built-in adjuvants if required, which can help to penetrate or can help to retain a spray on the spray droplet on the surface of leaf. But again, there are limitations of OD formulation, which we know difficult to formulate, difficult to develop and stabilize, can suffer from sedimentation, which we have seen a picture in previous slides. So again, these are the limitations of OD formulation. But then you have to find out a way how we can work and get a best formulation which will be giving good efficacy and good shelf life. So what we did a simple experiment that we mixed microbes, here in this case trichoderma, with metaspore 550S in one case and with inert in second case, and try to find out which is behaving better. So in A you can see pure conidia, say trichoderma alone. In B we have conidia blended with inert material, and in C you can see that that conidia is blended with inert material and 550S. You can see 550S is working well in this particular formulation. Then we did a viability testing of some of the formulations. So what we did, we initiated some studies of few of our products which are there in our basket to understand how these can help to helping to you in selecting these components while developing your formulation as WP or OD. Now what we did with trichoderma, we used is as a model, and we assessed it with our various products under stress conditions to highlight the performance of the combination of the two. And we did two complementary methods we used here to assess the viability. One is CFU, that is colony forming units, colony counting, also known as, and then second is direct viability through conidia germination test. So these two methods we have developed. Basically, this compatibility of Croda products with microorganisms, that is recent method that developed by us, this quantitative measurement, and we did this for Bacillus thuringiensis also, and then this method is helping us to fast understand the compatibility of microorganism and Croda products. And we are also working with various other microbes like Pseudomonas, which can give a compatibility with some of the Croda products. So this is what we are working on. So what we did in this particular study, we mixed all these ingredients and we stored at 20 degrees centigrade and 25 degrees centigrade, and we analyzed this over 180 days. So what we found during the analysis is that you can see here, so you can see extreme left is control, then you can see Croda 1, 2, 3, 4, 5, and 6. So, 6 samples from 0 days to 180 days, and you can see how, for example, compare control versus Croda A. You can see that after 181, after 30 days or 60 days, performance is still better. So each component can affect differently to microbial viability, that you can see here. Here we have used trichoderma species and conidia viability. We checked over 180 days. Then what we did, as I mentioned earlier, that purity grades of surfactants also help to improve the viability, shelf life, etc. Here you can see that what we did, pure conidia we took. Second is standard surfactant, and third is high purity surfactant. same surfactant standard, we did purification and use that. And you can see the difference. In case of standard surfactant, you can see it's 26% viability, whereas in case of pure, we can get 54%. So that is what the effect you can see on microorganisms for viability. But of course, here we have checked with trichoderma, but when you have the different one, you have to test it at your end, or you can ask us to give or do some testing for you, and definitely that will help to generate some data for you at our end, and we can work together on that. What we did for the viability test, we did different technologies for maintaining viability and evaluation. What we did here, we can see the trichoderma again, high grade surfactant we put in A and improve purity of surfactant. What we found in earlier slide, same we have seen under microscope. You can see that red arrows, what we have showed here, they are viable conidias or activated non-germinated or germinated one. And these pictures we have taken under microscope, which is there available in our laboratory. Viability test, again, we tested two different Croda surfactants, Atlas Metas for 550S and Atlas G1086. Now, both are well known to you all. And again, we did a study with trichoderma. And what we found that you can see pure conidia versus when you add 550S or 1086, you can see the difference in viability test of trichoderma. So definitely 550S and 1086 are similar behavior when compared to the control. Theoretically, the use of these products can reduce the natural drop in conidia viability over the time. So that's what we conclude from here. Formulation stability test, I'll not go much in detail because you know what methods are people are using, what type of formulations are there. And then microorganism survival assessment within full formulation development, we can do a viability test. And then we are developing few next steps or what are there under development. Now I'll take you through the Croda products, what our solutions or recommendations to all these biological formulations. Croda portfolio, what we have is we have 100% biological based products, renewable energy used, and then same performance as petrochemical products. So we have eco range products. At the same time, we have OMRI listed products. We have some of our products are listed at 4B list of EPA as well as OMRI certified products. And these products are proven solutions for adjuvants or additives or in-seed treatment. So this is what we are having in our basket. Few of the products we are showing further. At twin range, as I mentioned previously, this twin range or this EO based or natural EO based twin, excellent option for as emulsifier for biological formulations, enhances surface activity to provide better wetting agent. Atlas 4916 is superior interaction based type of star polymer type of product, and it has superior anchoring that will give a stability to the formulation. Oil-based formulation, basically it's OD. Atlas G5002L, again, effectively as aqueous dispersion for the microbes upon dilution in spray tank. So this bottle is 100% active, water-free, so can be used as adjuvant in spray tanks and suitable for sensitive microbials at low use level. So if you have moisture sensitive microbes, you can use Atlas G5002L. Atlas Metasphere 550S, we have seen the examples. This is a polymeric dispersant, and we have seen that in our studies gives very good results with trichoderma. So again, this is a powder form, can be used in WP or WDG type formulations. Can also be used in OD as aqueous dispersant. When you add your OD formulation in water, obviously you need some dispersant there. Atlas G1086, we have seen the example. This is emulsifier for methylated seed oil and vegetable oil, has low phytotoxicity and which is good for the viability of the microorganisms. Rheostux 100 and Rheostux 200, these are rheology modifiers for oil-based formulation, enhances stability, improves the viscosity of the formulations. So Croda can collaborate on projects or develop new partnerships or share our knowledge to make your formulation development as simple as possible. So please do consider Croda as your partner for developing biological formulations. With this, thank you very much. And if you have any questions, please let us know.